Hi, my name is Arianna Krinos. I'm a graduate student in the MIT Hui Joint Program, and today I'll be talking about daily patterns and expression in Western Antarctic Peninsula metatranscriptomes using a metatranscriptome analysis and assembly pipeline called Eukrhythmic. And the Western Antarctic Peninsula is particularly important to study because it's warming dramatically. In recent years, there have been extensive warming recorded of up to 8 degrees Celsius. And the phytoplankton community is particularly important here because it supports higher trophic levels through the feeding of krill in the region. There's a succession in the composition of the phytoplankton community over time in the Western Antarctic Peninsula, starting from a diatom and haptophyte-dominated community and progressing to a more cryptophyte-dominated community. I was interested in exploring the role of taxonomic composition in function and daily rhythms observed in the Western Antarctic Peninsula, as well as whether these patterns are conserved within a day and between different regions of the area. And in order to do this, I used samples from a 2014 cruise in the Western Antarctic Peninsula, which explored this region and its biology. In particular, taking a series of metatranscriptomic samples from the points labeled on this plot, with a series of diel samples taken at the location labeled in blue over the course of a 28-hour period in 4-hourly intervals. And as you can see here, the temperature variability was low within a day as labeled on this plot with the blue box. Within the diel samples, CTD profiles of temperature were relatively constant. However, in different subregions of the Western Antarctic Peninsula, there are markedly different temperature profiles. In this region, we also expect to see relatively low variability in light availability, in particular in the summer months, as well as the winter months close to the solstice. However, as you can see from this plot, as an average of photosynthetically active radiation in the first 50 meters over the course of the cruise on the left, and specifically within the diel data points on the right, there is significant daily variability in light availability in this region. And by using assembly with Eukrhythmic and annotation with the compatible tool Ukulele, I was able to derive the taxonomic identity of each of the contigs found in the metatranscriptomic assemblies as shown here. And when annotating these contigs and counting them up solely on their annotated identity, we can see that the community tends to be dominated by dinoflagellates, which is the green bar, and particularly in the diel samples. However, when we look at abundance weighted communities, as shown here for the diel samples, we can see that the community was actually dominated by a haptophyte, in particular in this case by a bloom of the haptophyte Phaeocystus antarctica. And using the assembled and annotated contigs from Eukrhythmic, I annotated them taxonomically with Ukulele and created groupings, taxonomic groupings of each of these sets of contigs with compatible reference genomes in order to conduct orthologous group clustering to identify functionally similar groupings of genes to track their abundance over a day and between regions. And this process entailed taking different taxonomic groups, including chlorophytes, centric diatoms, cryptophytes, dinoflagellates, haptophytes, and pinnate diatoms, and exploring their daily variability in expression. And this process yielded different numbers of orthogroups for different taxonomic groups. Here, the red bars correspond to orthologous groupings that contained a sequence both from metatranscriptomic contigs as well as reference sequences or sequence. The green bars correspond to orthogroups containing only a sequence from the metatranscriptomic contigs, and the blue bars only the reference. You can see for the dinoflagellates, as compared to other groups, there was a relatively high number of orthogroups that contained only a sequence from metatranscriptomic contigs, whereas for something like the pinnate diatoms, a high number of the sequences from the reference genomes were covered in these orthologous groups with metatranscriptomic contigs versus being contained only within their own orthologous grouping. And this corresponds to the high diversity of the dinoflagellates in comparison to other groups. The dinoflagellate group that I was using in this analysis contained the highest number of distinct taxonomic orders. And when we look at diel variability as assessed by the non-parametric diel pattern detection software RAIN, we can see that there's also differences between taxonomic groups in terms of the number of significantly diel varying orthogroups that were recovered, with haptophytes, dinoflagellates, and cryptophytes having the highest number of distinctly diel varying orthogroups, whereas pinnate diatoms and chlorophytes tend to have low numbers of diel varying contexts. And we can explore each of these sets of diel varying orthologous groups, in this case shown for the haptophytes, and we can look at the functional annotation of these groups. In particular, we can see that at 9 a.m., orthologous groups that have annotated functions as calcium ion binding and carbohydrate metabolism tend to peak, whereas as we go through the course of the day, these orthologous groupings tend to be more related to digestion and transport, whereas early in the morning at 5 a.m., light harvesting tends to become important. So using this kind of a workflow, we can begin to explore diel changes and functions